underlying tauopathy. She completed her PhD at Cardiff University and then moved to New York to the Icahn School of Medicine at Mount Sinai. And then in 2022, she moved back to the UK, the Dementia Research Institute at Edinburgh University to start her own lab. And Kat's lab integrates functional genomics, biochemistry, and iPSC organoid models, which I'm sure you'll explain, to investigate the regulation of MAPT expression, function, and splicing, and how these processes contribute to tauopathies. So many thanks for joining us today, Kat, and I will hand over to you.
Super, thank you. That actually is my first couple of slides. Don't need to go over. The usual question, can you all see my screen?
Yes, we can, thanks. And
the presentation, right? Okay. Okay, super. So yeah, thank you very much for the introduction to come and speak to you all. I did want to include a couple of slides to just let you all know who I am, as I've not been in this group before. But today I'm going to give you a couple little snapshots about things we're doing in the lab, using stem cell models to try and understand how tau is becoming dysfunctional and not working properly in PSP and frontotemporal dementia, FTD. So just to reiterate what Rowena has already kindly introduced, I actually realized when I was putting these together that I started my undergraduate degree 20 years ago, which came as a bit of a shock. So I actually started off in psychology at the University of Nottingham. When I moved to Cardiff University, I worked with Leslie Jones. This is actually where I studied Huntington's disease. And this is where I learned how to grow cells, how to do molecular biology, how to pet, and all those kind of fun things in the lab. And then I spent about 7.5 years in New York at Mount Sinai with Alison Goat. And that's where I fell in love with everything and anything tau. All of my projects were purely focused on tau, and that's where I learned how to do stem cell work and genetics. So that brings us to Edinburgh and now. So the lab was started in September 2022. So we're coming on almost 2.5 years now of me being independent. As Rowena mentioned, we're within the UK Dementia Research Institute, the UK DRI. So if you're not aware of it, it's a national institute that's made up of multiple different centres around the country that take a very multidisciplinary approach to studying dementia. So we have clinicians, people working on translation, and also basic biology like me. And the focus of my lab is largely on using stem cells to model primary tauopathies. And this is mostly PSP and FTD. So I wanted to start off by explaining why we use stem cells and what stem cells are. So obviously we're interested in the brain, given that we're studying neurodegeneration and dementia, but the brain is incredibly inaccessible compared to many other organs. So we can't just start poking around in there in people who are living. We can't manipulate how things are happening very easily. And the brain is also a very complicated organ. It's got lots of different cell types in there. So in approaches that we can use to look at the brain, like imaging, like MRIs and PET imaging and stuff, we can't really tell the difference between what different cell types are doing. We can't really figure out what's happening on the molecular level. So we need to find a different way to understand how brain cells are behaving or how they're behaving wrongly outside of someone's actual head. So to do that, we use stem cells. So what we can do is we can take, let me put a pointer on. So what we can do is we can either take a skin biopsy from someone, usually from their upper arm, or a blood draw, and we can take skin cells from that biopsy called fibroblasts, or we can take blood cells called PBMCs. And what we can then do is turn those cells into stem cells. So that's the induced part of the induced pluripotent stem cells. We're not starting with stem cells. We're taking a different cell and turning it into a stem cell. And so what's so powerful about these stem cells is we can now turn them into any cell in the body. They have the potential to become anything. So that's what the pluripotent refers to. So because we're interested in the brain, we can then take these stem cells and we can turn them into a whole host of different cell types that are found in the brain, as well as some more complicated kind of 3D models. These are the organoids. So in this talk, I'll just touch very briefly on projects we're doing on each of these cell types and sort of what the organoids are. But first, I just wanted to explain a little bit more about how we can use iPSCs to model PSP and FTD. So we leverage the fact that there are very rare cases where there's a mutation in the gene that codes for tau that actually will cause a mutant tau and cause FTD. So MAPT is the gene that encodes for tau, and in some people, the DNA has an error in it, which means that the tau is not formed properly, it isn't spelled correctly, essentially, and it's not functioning properly in the cell. And those people will get FTD. It's something called autosomal dominant. So if you inherit this mutation, it's guaranteed you will get this disease. So what we can do is we can take those skin biopsies and those blood draws from those patients, and we can make iPSCs from their cells. So now we have stem cells that have a mutant tau that we know is disease causing. So that means we can then turn these cells into any different cell in the brain, and we can see how that dysfunctional tau is having an effect. We can then go a step further and we can use a process called genome editing to correct that mutation. So we can essentially take out the misspelling and we can paste over it with the correct spelling. And this now means we have cells with a normal, healthy tau that doesn't cause disease. And so this is mimicking what we call controls, where this version of a healthy tau would not cause FTD or PSP. And so the real power of this is that we can actually now model disease and health actually in the exact same individual, because our tau mutation cell and a healthy cell are genetically identical. They're from exactly the same person. The only difference is whether that tau mutation is in there or not. So that gives us really good power to find things that are ever so slightly different with the presence of disease, or in the case of a healthy cell. So before I move on to the next bit, are there any questions, first of all, about iPSCs or stem cells or this approach?
I think you've described, you've done a pretty good job of describing it. I suspect some of us will have to reflect on it, and
percolate, and then
yes, it'll probably
okay. No problem. I'm perfectly comfortable if anyone ever wants to interrupt, just shout. If something doesn't make sense, that's also completely fine with me. Okay, so I'll tell you the first little snippet of something that we're doing in the lab, focusing on neurons, first of all. So this is a picture of some neurons we've made from iPSCs. So the blue is showing you the nucleus of the cell, and the green is tau, actually. So you can see tau in all of these long projections of the neurons. Neurons are all reaching out and connecting with each other so they can communicate with each other. And this is the primary function of neurons in the brain. So they need to cover long distances so they can talk to each other. They send signals around the brain. They essentially, they're essential for how you think and feel and move and act. And to do all of these processes, you need a lot of energy. So the way cells produce energy is through mitochondria. You may have heard of mitochondria being referred to as the powerhouse of the cell, or as the battery of the cell. I'm going to add another analogy and say they're like the power plant of the cell. And so what they do is they take in oxygen and they convert that oxygen into energy, or ATP. And so we wanted to see whether this process might be different in our mutant neurons, and it may explain why they function differently. So we carried out this assay to look at the amount of oxygen that mutant neurons are able to consume. And just to let you know, whoever invented this called it a seahorse assay because they think the shape of it looks like a seahorse. I would suggest they were wrong, but that's what this is called. So what we can see is the amount of oxygen that mutant neurons and control neurons consume over time. These are just drug treatments that make the mitochondria behave differently. You don't need to worry about those. But what you can see is the amount of oxygen is higher in the mutants in blue and orange consistently over time compared to the controls. But when we did all the maths and we kind of looked into this assay a little bit more, what we found is that the mutants weren't actually producing any more energy than the controls. So this is the amount of energy that the neurons are able to produce. So what seems to be happening is a lot more oxygen is needed to go into this reaction when you have a tau mutation or a mutant neuron, but it's not increasing the energy output. So that suggests that the mitochondria aren't functioning properly. We can then look to see, well, how many mitochondria do the cells have? Maybe there's just a difference in how many are there. So we can do this assay to look at mitochondrial mass. And this is now in the mutant neurons compared to controls. So if everything was the same as a healthy cell, the mass would be at 1, where this pink line is. And I think you can hopefully appreciate that mass is much lower in the mutant neurons. Now I'm saying mass because with this assay, we can't actually tell the difference between whether mutant neurons have fewer mitochondria compared to the controls, or whether they just have much smaller mitochondria compared to controls. But in either case, they're not able to perform this function very efficiently. Finally, we looked at something called reactive oxygen species. So this is ROS. And ROS is actually a byproduct of this reaction. It's a normal byproduct. It's supposed to happen. And normally, healthy cells are able to get rid of that ROS very easily. But when it builds up, it can actually be damaging to the cell and it can cause some damage. And so what we found in our mutant neurons, again, the controls are this pink line, is they start off when they're very young, fine. The amount of ROS is the same as in the controls, but as the neurons start to age, the ROS production gets higher and higher. And that accumulation is actually toxic. So you can kind of consider it an accumulation of toxic waste due to these mitochondria needing to work harder. So the summary is that what we're finding is that in mutant neurons, mitochondria need to work harder to produce the same amount of energy, probably because they're smaller or there's fewer of them. And that process is creating a harmful waste that's then damaging to your neurons. So this still leaves the question, well, why does mutant tau affect mitochondria? This is something that we're working on in the lab at the moment. I don't have the answer yet. But we and others have shown that tau should interact directly with the mitochondria. So in control cells, you do find tau in mitochondria. And we think that might be because the tau helps the mitochondria move around the cell via these things called microtubules, which are essentially like the skeleton of the cell. And so in the mutant case, the dysfunctional tau is not able to be on that microtubule as it's supposed to be. It's no longer interacting with the mitochondria. And so it may be that this mutant tau is not moving the mitochondria around the cell in the way that it's supposed to. So then they're not being regulated properly. And so they're not working very efficiently. And we have seen that there is a difference in how tau binds to mitochondria if there is a mutation there. So we can actually isolate the mitochondria out of the neurons. This is a protein representing that we've isolated mitochondria. And when we look to see if tau is present, it's only there in the control neurons. The tau is no longer associated with the mitochondria in the mutant neurons. So this is something that we're still spending a lot of time working on. We want to try and understand, well, why is it not interacting? Why are the mitochondria working much harder? And so hopefully we'll figure that out. And then the idea would be if we can interrupt that process, right? So that we can get mitochondria functioning properly again to keep neurons healthier. So that's kind of all I was going to say about neurons. If anybody has anything they want to jump in with at this point.
So there's a question, I don't know whether it fits here. Are you looking at epigenetics?
I'm not actually, no, that's probably one of the things in the lab we don't look at. So we do a lot of genetics and a lot of transcriptomics, like in terms of how genes are expressed and how they're modified, but not epigenetics, no.
And do you just want to, while we're on it, tell us what epigenetics are?
Yes, yeah. So epigenetics is ways in which your DNA is modified, essentially, so that can affect gene expression. So it could be anything like the way that DNA is folded in the cell. So it has to DNA isn't in your cells in like these long strands. It's all clumped together in all these complicated structures. And so the shape of that folding and how efficiently it's folded, that can change how your DNA is used. It can also have different things added to it that affect the way genes are expressed. So there's something called methylation, where a methyl is added to particular parts of your DNA, and that can either turn genes on or switch genes off. So it's a way that your cells regulate how they function that's not necessarily inherited. So a lot of these modifications can come from environmental exposures and chemical exposures. So it's like an additional level of regulation above just your DNA sequence.
Got it.
Does that make sense?
Yes, and so it's something else that can affect what's going on in the cells effectively.
Yes.
Yeah.
Yes. Yeah.
Okay.
Just to make it more complicated, basically.
And I've got another one. Are there any current drugs that improve mitochondrial function in the cell?
There almost definitely will be. The problem will be whether they cross the blood-brain barrier, or whether they're targeted to specific cell types. So one thing about the work that we're doing in iPSC models is that you have the ability to isolate individual cell types, right? So we're only looking at neurons here,
Yeah.
for example, but we don't necessarily see mitochondria dysfunction in other cell types. So it might end up having to be a more, I suppose, targeted therapy somehow, so that you're only affecting mitochondria, because you only affecting mitochondria in neurons, because you might not want to hit it in other cell types. So a drug's just going to kind of go everywhere. So we might need to develop something more nuanced somehow. But yes.
Okay, I've got a few questions here about mitochondria.
Okay.
From Phil, are they specific to certain cell types?
So mitochondria are in every cell. Yeah.
Great, thank you. From Quentin, is there a timeline for answering the mitochondria question you're currently working on?
Well, grant funders hope so. So, I suppose you're probably referring into, how we can get from mechanism to therapy. That's always the big question that's looming over us. It's really hard to estimate, because until we've figured out exactly what's happening, we don't know how complicated it is or how easy it is to target. I would say for general pipelines or timelines for going from basic science to clinical trial, you're probably looking at something like 15 to 20 years, because we're at the very early basic science stages, right? So we're finding mechanism. Once we found mechanism, we need to find some kind of target that we can direct drugs towards. Once we've done that, then we need to go into preclinical studies. So that's probably adding it into mice, for example, to make sure there's no toxic effect of what we're doing. Once you've done that, you need to go through, all the safety trials and things. So it's, even if we are successful in figuring this out in the next 3 to 4 years, there's still a lot more background work that needs to be done before this can get to clinic, essentially. So it's difficult, because the work we do sometimes feels very far removed from patient populations, even though that is where we're ultimately aiming for.
Yes, yes, absolutely. And then Paul is asking, did I hear you correctly when you suggested that tau imperfections anomalies can be hereditary?
Yes. So we call them tau mutations. It maybe seems a little bit harsh to call them mutations, I suppose. They are very rare. So, but they are in the DNA. People are born with them, and they get passed down through generations. So what typically happens is you have these sort of small families or kind of regions where there's maybe been, a lot of not so much mixture between different people, where you have sort of little clusters of people with the mutations. But the vast majority of people don't have a mutation. The power of using them in the iPSCs is because we know that that cell is going to be unhealthy. Whereas if we took cells from someone who doesn't actually have a mutation, but that might be very complicated genetic factors that lead to them having disease and environment, that's actually very difficult for us to model in a dish. So using the mutations gives us a lot more power. And the assumption is that whatever is going wrong in the mutation cases is the same as what's happening in people that don't have mutation.
Thank you. And would I be right in thinking, because you did talk specifically about the misspelling that leads to frontal temporal dementia, that when someone presents with symptoms, that would be something that would be tested quite early on?
Yeah, I mean, if there is a family history, absolutely.
Okay.
Yes, it's very easy to test for.
Okay.
I don't work in clinic, so I don't know
But
the
lots of people.
procedure exactly how that decision is made. But yes, that would be tested very early on, especially if there was a family history of similar uh, disorders.
Great. Thank you. Well, you've answered the questions for now, so we'll let you get back to your.
So yes, thank you very much for the questions. That was really useful. So the next couple projects, they're much smaller. They're still very much in development, but we're going to talk about some different cell types. So the next cell type that we make in the lab is astrocytes. You can appreciate they look very different to the neurons. Their shape is very different. My husband saw this yesterday and he said it looks like koi fish in a pond. That's what he thinks they look like. I think they're very pretty. Astrocytes, as well as looking very different to neurons, have a very different function to neurons. They play a very important supportive role, so they help the neurons support their structure, they support them in their communication with each other. But astrocytes also have a role in inflammation, so responding to things that are going wrong in their environment. It's a similar process. You can imagine it as a similar process as when you cut your finger and the area gets swollen because lots of cells move to that area to try and repair the wound that you have there. It's a similar thing with astrocytes. So they detect something that's wrong in their environment, maybe a bit of a dead or dying cell or a protein that's not supposed to be there, and they'll respond and react, and they'll rush over to that area to try and fix it. And so we found that levels of, we call these inflammatory cytokines, but they're essentially these proteins that the cells throw out when they're in this reactive state, when they're finding that there's something wrong in their environment and they need to have that inflammation reaction. Mutant astrocytes have much more of that just when they're sitting around. So we're not doing anything to them, we're not stressing them out, they're just sitting in a dish. But just having that mutant tau is making the astrocytes more stressed. They're already much more reactive just because that mutant tau is present. So the other thing is that, as Zane already introduced, this tau accumulation in astrocytes is a diagnostic feature of PSP. So this is an example of an astrocytic tuft. So we wanted to see if we could model this in our iPSC astrocytes. So what we did is we can take tau protein, we can label it with a red fluorescent dye, and then we can add it onto our iPSC astrocytes, and then they'll gobble up that tau. They'll just eat it and they'll take it inside themselves to try and clear it away, and that will cause the cells to glow red. And so what we found is that when we add tau onto control astrocytes and mutant astrocytes, they gobble up the tau really quite quickly, but the mutant astrocytes eat it much more rapidly than the control astrocytes do. And that might be something to do with them already being in a more reactive state, but they take up a lot more tau a lot more quickly. And then what we found is that if you actually look at what kind of tau is now in the astrocytes, we can detect this kind of clumped up aggregated tau. So the astrocytes are taking up tau from their environment, and instead of processing it and getting rid of it like they should normally do, they're actually forming it into these little clumps. And that could be a precursor to the formation of these astrocytic tufts. So what we're seeing is in the mutant astrocytes, not only is tau eaten much more quickly, but it isn't getting broken down properly, and that may be leading to the astrocyte pathology, as Zane was discussing. That's all I have on astrocytes at the minute. So I'm going to skip over to microglia, which is another glial cell type in the brain. I don't have a beautiful picture for microglia, unfortunately, but this is what they look like when they're growing in the dish, just down the microscope. They are, as you might expect, very small, in micro, in fact. And they, again, have a very different function to astrocytes and neurons. So microglia are like the immune cells of the brain. Like astrocytes, they surveil their environment, they pay attention to what's happening, and they go and they respond by eating up anything that's not supposed to be there. So bacterial particles, maybe in the brain, or viral infections, little bits of broken cell, proteins that aren't supposed to be there. And they essentially need to take care of the brain environment so that all the other cells can function properly. So what we're finding in microglia with the mutant tau is that, similar to astrocytes, they also eat things much more quickly that are in their environment. This time we didn't label tau with that red fluorescent dye. We labeled these bacterial particles with the red dye, because we know microglia respond to that very quickly. And so we see the mutant microglia eat that up much more quickly. The next thing that we wanted to see is, well, if there's a mutant tau in these microglia, are they responding to their environment differently? That's kind of the main thing that microglia need to be able to do. And so what we did here is that we had control microglia and we had mutant microglia, and we treated them with these different compounds that forced the microglia to react. And what we found is that, these are just some of those molecules that get pumped out of the cell when they're in a reactive state or when they're responding to their environment. So they're kind of markers of reactivity. So what we found is that in some cases, the mutant microglia were much more reactive to some of these stimuli. So this one is much more yellow. It's a higher level than the equivalent condition in the controls, and also down here. But there are also some interesting examples where the mutants weren't producing things that they should be producing. So for this molecule called IL-8, for example, it's missing from the mutant microglia just when they're sitting around, but it should be there in the control microglia. So we're still digging through this data and we're replicating it, but it's indicating to us that mutant tau microglia are functioning differently and they're not responding to their environment appropriately. So similar to the astrocytes, they do eat things much more quickly and they're responding very differently to stimulation. So to kind of tie those three projects together, I don't know if you've seen this meme, but it's all connected. It's all related. None of these things are kind of happening in isolation. But to summarize, what we're seeing in the neurons is that energy production is very inefficient, and that inefficiency is damaging the cell. And that means that communication around the brain is being affected. We then move on to the astrocytes, and if you have damaged neurons, the tau from those neurons is going to be taken up by surrounding astrocytes. That tau is then aggregating, it's affecting their function, and they're no longer able to support neurons. And then we have all of these things happening when neurons and astrocytes are unhealthy. That's going to cause the microglia to respond, but they're already in an overreactive state. They're not able to respond to things happening in the brain properly, and so they're less able to protect other cells in the brain. And so they all merge together, they all affect each other. And so what's nice about using these IPS systems where we look at each cell type in isolation is we can understand how each one is individually behaving differently, so that we can tie it all together and understand the whole process and the whole network of what might be going wrong in an entire brain. Okay, so I'm going to pause again if there's any questions before I just talk a little bit about organoids. So if anyone has questions about the glia part.
No new questions at this point.
Okay, super. Okay, so I'm not going to talk too much about the science for the organoids, because a lot of that is gene expression and it's very dry. So I thought I would just kind of use this opportunity to tell you what the organoids are, and show you some nice pictures of them, to get you excited, hopefully, about the model. So the work I've been describing to you is what we call 2D monoculture. So cells are grown flat in a dish and they're just one cell type, so that's the monoculture. And as I described, that can be very helpful, but it's also very reductionist. So it's nothing like the complexity of the human brain at all, which has got loads of different cell types. It's got this very complex layered structure to it through the cortex. We don't mimic that at all in the dish. So it's going to be the case that there are things that we miss, that might be related to structure or function of these other cell types like microglia, endothelial cells, oligodendrocytes, and so on. And so there is a model now that has been around for a few years that people are constantly improving, called organoids. So organoids kind of are starting to bridge this gap between 2D monocultures and actual human brain. So they're from iPSCs still, and they form these 3D tissues that have more complex mixtures of cell types in them. They have some representation of different cortical layers, but nothing as complicated as actual human brain. But they don't have every single cell type. So they don't have microglia, endothelial cells, oligodendrocytes. They don't have blood vessels. They don't have blood flow, things like that, unless they get kind of engineered in afterwards. But they do allow us to look in a more complex human model that's getting closer to mimicking the human brain. So they have some real advantages with that. So this is what they look like when you grow them in a dish. Quite disappointing. They're just these little sort of pink balls. And for a sense of scale, each one of these is about 2 millimeters in diameter. So they are very small. They're nothing like the scale of a brain. But when you look at them, with some antibodies and a process called immunofluorescence, they've got these really complicated structures and really beautiful integration of different cell types. They're a really cool, interesting model. So we're developing a toolbox of things to do with the organoids. We're trying to do as much as we can in an intact organoid without having to cut it, section it. We want the cells to be alive when we're looking at them so we can understand their function. So one of the things that we're doing is something called calcium imaging. So this is essentially a way to just watch how neurons are communicating with each other. So when a neuron sends a signal to another neuron, you get this influx of calcium. And we can detect that with this green fluorescent signal. So we can watch that in real time. We can see that this neuron is sending a signal to another one, and then that signal dies away. And then we can start looking at how fast that happens, how many cells are doing that, and we can see how that communication between cells is different between mutant and control. Similarly, there's a process called multi-electrode array, where we can do this. So it's another way of just measuring how neurons are communicating with each other. So we measure their electrical impulses. We can also do these very high-powered microscopy techniques. So this looks like it's a cartoon, but this is actually what a synapse looks like in an organoid. So this is where one neuron is connecting to other neurons, and all of these little tiny dots are filled with neurotransmitter that are about to be released. It's really cool. And we can also see the mitochondria at this level as well. So we're using this to see if mitochondria shape and size are different between the mutants and the controls as well. We can also do these other microscopy techniques called array tomography. So in this case, we have embedded an organoid in this little capsule, and then it's sectioned very, 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 very thinly. And we can visualize individual synapses. So we can see how that might be different between mutant neurons and controls as well. And finally, we're trying to make the models more complicated. So I mentioned that organoids don't have every cell type in the brain in them. One thing that we're interested in that I've not touched on yet today are oligodendrocytes. So we're developing models where we add those into the organoids, and we can track how they manage to integrate with other cells and how they spread out and what their shapes are and what their function is. And we can again see how that might be different between mutant cells and control cells.
I'm just going to do a quick, oh, great. I was just going to do a time check and you've got to your final slide.
Okay. Yeah, we can stop for questions now, or do you want me to just finish?
Just finish up, yes.
And yeah, so I appreciate the invitation to come and speak to you all. As I said earlier, with the basic scientists in the lab at the bench, we don't get the opportunity very often to actually talk to people. So it's always really nice to have the opportunity. This is my lovely lab, and always a huge thank you to all the funders, but also all of the people who have donated their tissues and their brains to this research that actually lets us do this work. We hugely appreciate that, and it's incredibly valuable. So yes, any more questions? I'll be happy to.
Thank you. Yes, we'll just do a few quickly. I'm conscious that Ed is on and ready to go, so I don't want to hold him up too much. But from Penelope, is there currently any way for a living PSP patient to donate skin or blood samples for research?
Yes, definitely. It would be to probably contact the nearest research lab and you can get it organized. But yeah, definitely.
So I would suggest you can do that via the PSPA, Penelope. We'll make sure we have the answers to questions on that. Mervyn, how does the work you're doing relate to the ongoing clinical trials on stem cells, such as treatment of for progressive for PSP with autologous adipose tissue-derived, wow, this is a mesenchymal stem cells.
I don't know. I'm not familiar with that trial, so I'm not sure. Are
Okay,
they
well,
using the stem cell? Are they replacing cells with stem cells, or are they using the
let
stem
us take
cells?
that one away, Kat, given the time, and we'll see if we can get an answer for Mervyn. And then one final question from Max. Please, can you suggest how any cure might be derived from the mending of mutant tau?
Yeah, so it's tricky. So there is the possibility for those rare cases that have the mutations that if that mutation can be identified, I mean, it can be identified at any point, it can be identified in utero. You can use genome editing in living people to correct that mutation. That's being done for very rare, I think, ataxias, to essentially cure them. It wouldn't, it's not an easy approach. It's not, it would need a lot of development and optimization, but people are definitely working on that. It isn't useful for most people with a tauopathy because, as I mentioned earlier, most people don't have a mutation. So there are trials at the moment using things called ASOs, so antisense oligonucleotides. They target tau and they just reduce the levels of tau that are in the brain. So the idea being that if the tau is not there, it can't aggregate, it can't cause problems. Those seem, as far as I'm aware, seem to be going quite well and having very positive results, but it doesn't change the tau so much as just reduce the tau. Our approach is to not target the tau directly, but to try and understand where else in the cell is going wrong. And that's what we're trying to target, what we're trying to fix, because we're probably going to need a multifaceted therapy. So we probably can't end up targeting just one thing. We'll probably need to target maybe some mitochondria here. Maybe we need to block astrocyte reactivity at the same time, so it's going to be a multitude of different therapies, maybe mixed with also reducing tau.
Great, thank you. Well, just a final, many thanks for joining us and for intriguing us with some of these new terminologies. I think these organoids are fascinating, and it does sound like they will enable us to do things a lot more quickly and in different